Hi everyone, my name is Alexis Uria. I did my presentation on chemoreception, the senses of smell and taste. Starting with olfaction, I'm going to go over the basic pathway. So olfactory epithelium lines the nasal cavity and is embedded with olfactory sensory neurons. The olfactory nerve, which is also referred to as the cranial nerve 1, is made up of the axons of the sensory neurons. This nerve then synapses with secondary sensory neurons that are located in the olfactory bulb. The olfactory bulb here contains secondary higher order neurons, and these project outward through the olfactory tract that lead to the olfactory cortex. So, signal transduction occurs in several steps in the olfaction system. Olfactory epithelium is lined with a mucus layer that's embedded with non-motile cilia. So, odorant molecules must dissolve in and penetrate the mucus in order to bind to the olfactory receptors, which are G-linked membrane receptors here on the cilia. So these odorant molecules plus their associated olfactory receptor activates the G protein Gulf. Gulf then increases intracellular CAMP, which opens up CAMP-gated cation channels. This then depolarizes the cell, which triggers the action potential that travels along down the axon to the olfactory bulb. Once in the olfactory bulb, this contains secondary neurons where axons of the same receptors converge. These then modify the information before it is sent to the olfactory cortex. Finally, once in the olfactory cortex, the information is integrated to create our perception of different smells. This different sensory information can then be sent out from the olfactory cortex to the cerebral cortex and limbic systems for associations to be made. Just to go over the diagram of the olfactory bulb, I want to point out that the olfactory bulb presses right on the underside of the frontal cortex in the frontal lobe. Here you could see the olfactory bulb has many of the synapses from the dendrites that come from the olfactory epithelium. And from the olfactory bulb down the axons, the signal travels through to the secondary sensory neurons. So just to complete the olfactory cortex, we can see that it's right here, right under the gustatory cortex, which we're going to go through next. So gustation is our sense of taste, and it's actually linked to olfaction. It's a combination of 5 different sensations that have been identified as sweet, sour, salty, bitter, and umami. And each of these are associated with a unique physiological process. First, we're going to start with the basic pathway. So taste receptor cells are non-neural polarized epithelial cells, and these are embedded in epithelium in the taste buds. They poke through a tiny little pore called a taste pore. The membrane of taste receptor cells is covered with microvilli to increase the surface area and increase the contact with the environment. So one taste bud on the tongue actually includes 50 to 150 different taste receptor cells and support cells and regenerative basal cells. Each taste bud also has 4 distinct types of cells. There's type 1, type 2, type 3, and type 4. So a substance dissolves in the saliva and mucus of the mouth. Then one taste ligand interacts with apical membrane proteins on the taste receptor cells, which initiates signal transduction, which we'll go over in more detail on the next slide. Chemical signals are released from the taste receptor cell, and primary gustatory neurons are activated, and their axons run down through the cranial nerves and synapse in the medulla. Sensory information then passes through the thalamus and reaches the gustatory cortex finally, which is located in the insula. So to go over taste transduction in more detail, it's going to be separated into sweet, bitter, and umami tastes in their transduction pathways, and then salty and sour. So sweet, bitter, and umami tastes are sensed through the activation of G protein coupled receptors. Type 2 taste receptor cells recognize these tastes and express G-coupled protein receptors on the surface. So once the special G protein Gustation is activated, it activates a signal transduction pathway that first releases stored calcium and then opens cation channels that allow calcium through. Calcium initiates the release of ATP from the type 2 cells through a wide pore channel, and then ATP acts as a paracrine signal to sensory neurons and neighboring presynaptic cells. This communication causes complex interactions. Next, we're moving on to salty and sour tastes. These use ion channels. So first we're going to start with sour. So sour is recognized by type 3 presynaptic cells. Increasing H+ is known as the sour signal. So H+ mediated depolarization of presynaptic cells causes the serotonin release via exocytosis, and this excites primary sensory neurons, which then help recognize the sour taste. For salty tastes, type 1 support cells are utilized. So Na+ enters taste receptor cells via an apical ion channel, which then depolarizes the cell and initiates events that cause primary sensory neurons to fire an action potential. It's noted in our textbook that sour and salty taste transduction methods are not definitively certain, however, these are the proposed mechanisms. I made a basic little study aid here for you. It's a flowchart of the pathways of olfaction on the left. So just to review it basically, it starts with an odorant and a receptor protein, which then activates a G protein, Gulf. This then increases intracellular CAMP, which opens up the Na+ channels and depolarizes the cell. The action potential reaches the olfactory bulb, and then secondary neurons, which are located in the olfactory bulb, modify the information and send this out to the olfactory cortex where it's processed. On the right, here I have an empty diagram of the gustation senses. And if you go to the next slide, you can find the answers for the diagram and also another brief review of the pathway of olfaction. Thank you.